Hello and welcome to 2-Minute Tuesdays, brought to you by KCAS Bioanalytical and Biomarker Services. I'm Dominic Guarino, the Senior Scientific Advisor for Ligand Binding Assays and Biomarkers here at KCAS. Today I'll be talking about pharmacodynamic assays. Pharmacodynamic assays, often referred to as PD assays, are defined as the branch of pharmacology concerned with effects of drugs and the mechanism of action, or their MOA. PD assays are the study of biochemical and physiological effects of drugs, especially pharmaceuticals and biopharmaceutical drugs, on the body's biological response to drugs. Think of it as the PD assay is the study of how the drug affects the body or the organism, whereas the PK, or the pharmacokinetic, is the study of how the organism affects the drug. Both together influence dosing, the benefits of the drugs, and the adverse effects of the drugs. The most common questions that I get regarding PD assays are, what type of standard is used? So one of the biggest challenges with PD assays is frequently you use what's called a recombinant protein standard. And this is something that's synthetically engineered and is in the laboratory. And sometimes it does not have the same properties as the endogenous protein, meaning a protein that is naturally occurring within an animal or a human. So one of the challenges with PD assays is that when you have a recombinant standard, you frequently have to ensure that it behaves the same way as the endogenous protein. And so that can be done by procuring healthy donor matrix, but frequently requires patient matrix or even some tissue, depending upon the type of pharmacodynamic assay one is doing. One of the biggest questions we get is what type of regulatory requirements does it need? And this is often defined as the context of use. So will it be something that is a primary endpoint, something that will be using decisions on the drug's dosage, or is it something for inclusion or exclusion? All of those would require a full GLP. Alternatively, if it's in the exploratory research, sort of secondary endpoints, but that's a little bit grayer, that can be a non-regulated fit-for-purpose validation. But certainly in the case of something that is a pharmacodynamic marker, you would want some sort of validation. We at KCS have over 10 platforms to assist with your PD needs. We qualified or validated over 40 PD assays in the last 12 months, ranging from soluble cytokines and chemokines to activity assays in all sorts of different matrix such as sputum, probably about a dozen different tissue types across a variety of different animals and species, or something as sophisticated as a flow cytometric method, which requires a tremendous amount of effort and energy.